So I'm a researcher in Rennes, France, Institut NuMeCan, Nutrition, Metabolism and Cancer. And I'm not the Musso guy who works in diet or lifestyle. He's probably cousin. I've been living in France for 30 years. So I'm going to talk about fibrousness in hepatocellular carcinoma, which is a tumor niche enriched in Wnt signals and cancer progenitor stem cells. So, as you all know, hepatocellular carcinoma is associated more and more frequently with MASH and MASH, and go through all the stages of fibrosis, cirrhosis, and the emergence of preneoplastic lesions, and ultimately HCC. We recently described the inflammatory background in a muscle and MASH dataset that we built. The study was purposely designed to study the expression of the SARS-CoV-2 receptor ACE2, and we showed that the expression of ACE2 was related to collagen type 1, type 3, versican, collagen type 4, and laminin gamma 1 expression in these patients. We studied controls, obesity, patients with obesity, steatosis, MASH, and steatohepatitis. Interestingly, as we all know, ACE2 expression was related to age, but also to fat area, and the inflammation score was related to AC2 expression. Collagen type 1, 3, and versican, which are markers of molecular markers of liver fibrosis, were related to the fat area, and also these three extracellular matrix proteins were related to the inflammation score in the patients. We deconvoluted transcriptomic data to show that patients with steatohepatitis were enriched in native immune markers such as neutrophils, macrophages, dendritic cells, and natural killer cells. The decrease in KLRF1 marks and really an increase in natural killer cells. There was also an increase in B cells and T cells. As you all know, this inflammatory microenvironment in the across liver fibrosis leads to what is known as the field effect. The liver becomes a minefield where retrodifferentiation of hepatocytes and preneoplastic nodules appear. Hepatocellular carcinoma is a very heterogeneous disease, as you know, and this heterogeneity is seen at the macroscopic level. In most cases, on the CAT section and macroscopy, the tumor bulges over the CAT surface because the interstitial pressure in the non-tumor liver is much higher because there's, in this case, there's a cirrhosis, there's inflammation, fibrosis, which increases the interstitial pressure, but the tumors, HCCs in general, are soft masses with little or no fibrosis. At the microscopic level, these tumors are heterogeneous, and there's an array, a continuum of cytoarchitectural features going through from the hepatocyte differentiated phenotype with bicanalicular to less differentiated cells and the appearance of which would, in this case, would correspond to Edmonton score 3 and 4, with large nuclei, relatively small cytoplasm, and basophilic cytoplasms, and the appearance of some intratumor fibroblasts, which caught our attention a few years ago. So in parallel, we described, but first we created a metadata set of transcriptomic data from over 1,000 patients, which allowed us to describe a second dimension of hepatocellular carcinoma molecular heterogeneity. It was widely known from the beginning of the 2004 and 6 from Snorri Torgisson's work, that hepatocellular carcinoma heterogeneity was distributed along an axis, including the ratio of hepatocyte-like phenotype and progenitor-like phenotype. So the more retro-differentiated or the more undifferentiated the tumor was, the worse was the prognosis. Analyzing a large metadata set allowed us to describe the two poles of metabolic zonation for differentiated tumors. Among differentiated tumors, we could describe the perivenous one, which in most cases have a beta-catenin mutation, and the periportal ones, which display the default metabolic program of hepatocytes and do not contain beta-catenin mutations. So, hepatocellular carcinoma is also heterogeneous at the immunological level. This is a slide from the Joseph Lieuvet group published by the end of 2022 in Gut, where they describe different patterns of immune infiltration based on the convolution of also transcriptomic data, which may help predict the response to immune checkpoint inhibitors. Going back to intratumor fibrosis, what caught our attention was that despite the paucity of fibrosis in general in hepatocellular carcinoma, we found small deposits of intratumor fibrosis in some tumors, small hotspots about 5 millimeters to 10 millimeters in diameter, which allowed us to perform more molecular studies and to search for, if you can see my arrow here, to search for the phenotypes of the cells which were embedded in this nest, because as I showed you before, they tended to be to have a more undifferentiated phenotype. So we conducted sample study by tissue microarrays and analyzing mirror parallel sections from the same tumors by quantitative PCR, and we identified the first proof of concept that tumors with intratumor fibrosis were associated with progenitor cell markers such as CD44, LGR5, SOX9, also with secreted freeze-related proteins 2 and 5, which are associated with development of a myofibroblastic phenotype. Not surprisingly, we didn't find a positive correlation by correlation at all with keratin 19 and NuMeCan, because we had excluded from this first study, we had excluded hepatocellular carcinoma with a mixed hepatocholangio profile. Then by random forest, we identified a small signature which was associated with patient outcome. DKK1 is an inhibitor of the beta-catenin pathway. It was already very well known as a prognostic marker of hepatocellular carcinoma. So then we modeled intratumor fibrosis in vitro using Hepag cells by incubating these cells after replating different these cells. They go through 3-4 days transient mesenchymal competence, that is, they retro-differentiate to liver progenitors, and at that stage we treated them with Wnt signals, with soluble Wnt3a, and in 2 weeks we obtained myofibrous-like cells. These cells were subjected to a transcriptomic study, and we confirmed that they had lost the capacity to produce albumin, aldolase B, gamma GT, keratin 19, SOX9. SOX9 was quite complicated, the profile. We can talk about that later, NOTCH2, but they expressed progenitor markers such as LGR5, MYC, cervidin, BERK5, CD44, alpha small muscle actin, basement membrane proteins, fibrillar collagens, proteases, proliferation markers, and were able to invade matrix. Also, we identified a protein which was neo-expressed in very aggressive HCC and was the link protein, which form macromolecular assemblies in the extracellular matrix of tumors with other proteins such as versican and CD44. The conclusion of this part was that this phenotype associated with fibrousness could reproduce the bad outcome, vascular invasion, high alpha-fetoprotein expression, high prominent CD133 expression in hepatocellular carcinoma. So what we obtained in the in vitro and the transcriptomic study was confirmed in a small HCC series from our hospital of about 70 patients, from whom we analyzed 80 hepatocellular carcinomas. So as you can see here, HAPLN1, which is also known as the link protein, was expressed in the tumors, but also in tumor cells invading the stroma in an Indian file pattern, a pattern which is not very frequent in HCC except for very aggressive tumors, and which is quite frequent, for example, in breast cancers, in ductal breast cancer. As I showed you, CD44 and LGR5. So the first hypothesis was that Wnt signals were associated with mesenchymal commitment, the emergence of stem cell markers, leading to aggressive tumors with these markers, and this led us to propose the concept of fibrous nests. Obviously, we got quite interested in the extracellular matrix of this intratumor fibrosis. Extracellular matrix, as you well know, is composed of collagens, laminins, proteoglycans, and also growth factors. This extracellular matrix interacts with the cell surface and with pericellular matrix, which is a signaling platform. Here we can see TC44, which interacts with hyaluronic acid, and with the sugar polymers of glycosaminoglycans, and this is done by the link protein by HPL1 that, as I showed you before, also laminins, and the interaction of the extracellular matrix with the cell through integrins or DDR receptors. Also glypicans, there's not here, but we have significance here. So how to study specifically the extracellular matrix in human tumors, in human HCC resection experiments. The technique of the matrisome is basically a technique which goes through several steps of decellularization just to get the supramolecular assemblies of the extracellular matrix, which are then treated with succession of different detergents until we get really the extracellular matrix, and then is the classic digestion with trypsin and and then analysis by tandem mass spectrometry. So in this analysis, we studied 10 tumors matching non-tumor livers from the same patients. So with low-grade intratumor fibrosis and 10 tumors with high-grade intratumor fibrosis, we obtaining the low-grade fibrosis matrisome signature and the high-grade fibrosis matrisome signature, or which is called fibrousness. And we showed that low-grade was associated with non-proliferative HCCs with mainly oxidative phosphorylation metabolism and relatively good outcome, whereas the fibrousness matrisome signature was associated with a cancer-specific extracellular matrix, increased glycolysis, Wnt TGS-beta signaling, which is also known as the S1 Oshida subclass, and poor outcome. So we get networks of protein. This slide is just a little bit for fun, because when I was a PhD student in 1998, I had described the expression of collagen 18 in hepatocellular carcinoma for good prognosis, and we confirmed it here in low-grade fibrousness, preserving hepatocyte differentiation. Collagen 18 appears again in this context, which made us quite happy. This was done through a collaboration between Natalia Nieto lab in Chicago and our lab in Rennes, France. Romain Desert, working with Natalia Nieto. Romain Desert was my former PhD student who continued a 5-year postdoc with Natalia, validated this proteomics data with transcriptomics by integrating the signatures, using as a training set the metadata set that we had developed, and using a different validation cohort from TCGA. Basically, we described that low-grade fibrous nets are associated with transcriptomic signatures of good prognosis, where high-grade fibrous nets are associated with bad prognosis. The Oshida subclass S1, S2, and also with the T-cell exhaustion signature. Also, there's a difference in survival and different in the outcome of the patients and with a series of clinical features such as the metastasis signature, the periportal signature, alpha-fetoprotein, clip staining, Barcelona staining, tumor size, survival group, age, and the event of multinodularity. Obviously, we obtained a minimal signature, including several collagens, type 1, type 3, type 1 chain alpha 2, type 5, which also forms small important is very important, the relationship between collagen 1 and collagen 5, because collagen 5 is generally co-expressed with type 1 because it stabilizes the structure of collagen 1 fibers, fibrillin 1, NuMeCan, versican, periostin, EGF, and other small molecules. What is important is that this 11 gene signature was associated with a higher risk of death and recurrence in these patients. Then we localized using a multi or hyperplex technology from CellDive. We co-localize several of these proteins in hepatocellular carcinomas, and here we can see the co-localization of alpha smooth muscle actin. We used alpha smooth muscle actin, CD45 and CD31 labeling alpha smooth muscle actin, myofibroblast, CD45 leukocytes, and CD31 endothelial cells to have first a topographical landscape of the tumor. And then in the same slide, we localize the protein that we had identified by proteomics. This is elastin in magenta, periostin in turquoise, and fibrillin in kind of yellow-orange, which are forming kind of casts of around tumor nodules. These black areas are the tumor nodules surrounded by a dense extracellular matrix. And then we localize it in the same tumor, in the same slide, on the same slide, collagen type 1, dermatopoietin, collagen type 5, versican, also annexin 1, GDF15, CXL13, confirming their presence in high-grade intratumor fibrosis. So, in conclusion, high-grade intratumor fibrosis is associated with S1, S2 Oshida classes that we also call extracellular matrix as stem cell subclasses, because we found that it's very more transparent to call them extracellular matrix, because this HCC subclass, the blue one, is enriched in extracellular matrix protein, and this subclass stem cell is also rich in extracellular matrix proteins, but additionally express stem cell markers. So these two subclasses, intratumor fibrosis is found in general in these two subclasses. Also, by this proteomic analysis, we confirmed with what we had found a few years before and published in 2017, that the periportal HCC subclass, which preserves the default metabolic phenotype of hepatocytes, is the HCC subclass which has the best prognosis among all HCCs. We all know that HCCs has a very bad prognosis, but is the least, the least deadly subclass of HCC. This was confirmed in 247 HCCs from the Rossler dataset, and at that time on 210 HCCs from the TCGA dataset. So thank you very much for your attention, and here are the acknowledgements to funds, financial sources, and collaborators. Thank you very much.